As a reminder, so last time we'd finished working through the math of feedback, and we were looking at the caffeine feedback loop, and then so this is a commentary on the paper we'll look at. What goes on in the paper, so the experiment that these people did is they sort of had a feeling that this is what the pathway for caffeine activity was. And so what they do to test the hypothesis is they look at a mouse that has been knocked out for this protein to cut out this part of the signaling pathway. So if you look at the signaling pathway, there are the pathway downstream of the receptor goes to target proteins that they look at. They monitor the phosphorylation level. And there's also the path that goes through this protein DARP32. So by knocking out this protein, just this branch of the pathway is active, and then they get to see what this branch of the pathway is doing. And our prediction from a systems biology type model in the linear response regime is that without this branch of the pathway, we should still get activation of the target proteins. However, we don't have the positive feedback loop, so the gain might not be as great, and the duration of the effect might not be as great. So, so then we'll go to the paper. Let's see. All right. So this is from 2002, and so they say we know that caffeine blocks adenosine A2A receptors, but we don't know how it works. So what they did is they deleted DARP32, dopamine and cyclic AMP regulated phosphoprotein of relative molecular mass 32,000, and then they also looked at a drug that they know is an antagonist of the same receptor.
What does the molecular mass have to do with anything?
What does the molecular mass have to do with anything? The reason that the molecular mass is important is because that's how the protein was isolated to begin with.
Oh, okay.
So you put it, you fractionate brain, and you put it in a centrifuge to get different, different masses or sedimentations. Yep. Yeah. So that's the protein's name. It's because, a lot of proteins were named before we could sequence DNA.
Right.
So you name them based on activity.
The mass has something to do with the gain of the duration?
No. The mass has to do with the biochemistry of the way they found out this protein was important was they probably put like mouse brains in a blender and then looked for activities in different fractions. So here is, let's see, high resolution figure and legend. So in these figures, the regular mice, wild-type mice, are filled symbols, and the knockouts are open symbols. So they don't go to the coffee house. Instead, they get an injection of caffeine in A to C and G. So A, D, G, H, time course of the effects of caffeine on locomotion measured over 20-minute intervals. So we will look at A. So locomotion. So this is an experiment where, so let's see, what are the two locomotions counts per 20 minutes. So I think this first set of figures is like the mice getting acclimated to the cage. So they pick up a mouse, they put it in a cage. It takes about 100 minutes for the mouse to sort of calm down. So at first it's running around the cage, and then finally it settles down. So now the mouse is happy, all right? And then at time 120 minutes, they treat the mice with caffeine by injection. Then probably the activity like really spikes. They're not showing that. They start collecting data after, well, no, so they're collecting data every 20 minutes. So every 20 minutes they say how much did the mouse move the previous 20 minutes. These days maybe you get like a mini Fitbit to put on the mouse leg, but this is probably from taking a movie of the mouse and then having undergraduate researchers count how many times the mouse moves. So the immediate response, so looking at activity in 20-minute intervals, the response is the same whether the mouse has had caffeine or not. So it's just as stimulated. But then if you look at longer times, what happens is the wild type mouse in the filled symbols, the response is still greater, so the response continues. The feedback loop is active, so the signal stays on for longer. Whereas in the knockout, the signal, the output of the pathway is decreased, so the mouse doesn't move as much. And then in figure B, what they're showing is the area under the curve. So A is showing the time course of response. And it sort of follows an exponential, the way that all of our models have followed exponentials. Then the area under the response is also significant. In let's see. So then the results in D are treatment with something else that's affecting the pathway. And in G, or vehicle. So the results with D are from a drug that affects the same receptor as caffeine does. And so with this drug, they see a bigger difference. They see a difference at time 0 together with the difference in the area under the curve. But the same idea that getting rid of the feedback loop decreases the size of the response and also the duration of the response. All right, now let's see if there are any other figures in this paper that are going to be good to look at. I think that is the main one. So then this figure is, since you asked why is 32,000 important, like so here they're actually looking at the protein. They're doing blotting of, I think, phosphorylation activity to see that there's a change in phosphorylation of the proteins. So that's how the signal is carried through the network. All right, so we'll close that. And I think that's it for now for the screen. Okay. All right. So we finished the first big section of the course on linear response. And now we're moving to the second big section of the course on nonlinear biological pathways. And the focus is going to be on transcriptional regulation. And the two types of nonlinearities that we'll focus on in this section are saturation and cooperativity. So the biological picture is that we have a model for a cell, and here's the nucleus. And cells have receptors that sense the environment. They have other ways of sensing the environment. And we worked on a model for how signals get from the outside of the cell to the inside of the cell. They can change the cell's state by changing post-translational modifications on proteins. But they can also change the cell state by changing the proteins that are present in a cell. So that's sort of a longer lasting effect. And the way that happens is that transcription factors that are not active become activated, and they go to the nucleus. Here's DNA in the nucleus, and they turn on the expression of specific genes that then creates messenger RNA that's translated to protein. So these are the steps that we're going to look at. And saturation occurs because, so DNA is double-stranded, but we usually just draw it as one line. So there's DNA. Here's a gene. So say this is the gene that the transcription factor wants to activate. Usually, for most genes, there's only one copy of the gene per haploid genome. So humans are diploid. We have two copies of most genes, one maternally inherited, one paternally inherited. Other organisms are pure haploids, so most bacteria only have a single genome in them. Yeast cells that are used in the lab for experiments are usually haploid. So most genes we only have a single copy of, which means that there's only so much activity you can get out of a single gene. So if we think about, so say TF activity, so how much transcription factor is getting into the nucleus, and this is mRNA output. So it's sort of ignoring what happens at the low levels of activity. We'll think about that later. We'll just sort of draw something starting to go up. It might start out linear, but in the end, it levels off because we only have one copy of the gene or two copies of the gene. So there's just a physical constraint on how quickly polymerases can bind to the promoter, initiate transcription, and then transcribe the gene. You have to wait for the nucleic acids to get added to the growing chain, and it's just a physical constraint. At the next step of translation, there's also a physical constraint that there are only so many ribosomes, and they can only work so fast. There's only so much protein output that a cell can generate. For some genes, actually, this is such a problem. Having just two copies of the gene is such a problem that some genes exist in multi-copy in our genome. So most of our genes, we only have two copies, but there's some genes that encode proteins that are so high abundance in our cells, we need so many copies that there have to be multiple copies of the gene. So one example is histones. Histones are, so here, this is, sorry. So histones are core proteins that DNA wraps around. So the DNA in our cells is organized by wrapping around histones. So about something like about 140 base pairs of DNA makes a loop around a histone, like little beads that get wrapped around, and then that helps keep DNA organized. So we're not entirely sure of all the processes, but we know that there is a lot of histone, because for every 150 base pairs of DNA, we need a histone protein, and histone proteins exist in very high copy number in our genome. Another example of high copy number genes are transfer RNAs. So if you remember from molecules and cells, here we have a transcript, and there's a ribosome that reads off the codons and makes a protein, and tRNAs are adapter molecules that on one side have an anticodon that recognizes the three-letter genetic code, and then they're coupled with an amino acid, and we need a lot of tRNAs in order to decode all of the transcripts that we're making. So tRNAs also usually exist as multi-copy genes. But for the most part, genes are just 2 copies. So those are 2 exceptions. For the most part, genes are 2 copies in us, and for that reason, there's saturation in transcription. No matter how much transcription factor you have, usually you're limited in output. Yeah?
So do these multi-copy genes, do they not experience the saturation? Or they do just...
They'll get, yeah, it's sort of, so the way to think about it usually is, it's like evolution. That through evolution, we have copy number changes in our genome. And fitness pressure says that if there are too few copies, a cell or an organism will be lower fitness. So the copy number sort of increases until there's no longer a huge fitness penalty. So there are probably a range of copy numbers where sort of the fitness is equivalent. So most of us have other places in our genome where we have big duplications. We have copy number variation from person to person that is a big source of genetic variation beyond the genetic variation that often we think about in terms of mutations or variant alleles. Actually, most of us have aberrant copy number, well, aberrant, something other than 2 for a lot of our genes. So it's an evolutionary process that sort of has given each of us a range of copy numbers for many of these high demand proteins. Also, the pressure ends up, the fitness dependence on copy number for these sorts of proteins also depends on the cell proliferative state. So a cell that's trying to grow and divide very rapidly will have more of a fitness effect based on copy number. A cell that is sort of not really dividing, it's terminally differentiated, or just sort of hanging out, doesn't really have as much transcription translation going on, and so the fitness effects won't be as great.
Okay, thank you.
You're welcome. So that's saturation. The other effect is cooperativity, and that is sort of focusing on this part of the output curve. So in a linear system, no matter how small the input is, you get a corresponding proportional output. So at low inputs, everything is proportional. In a cooperative system, you often see a threshold. So here is input, and we'll think about this as a steady state input. So you're applying a step function, you're waiting a long time, and then you look at the steady state output. So this is the thing with the linear system. So the one we've been looking at, dx dt is equal to some steady state input minus decay. So if we set this equal to 0, we get that the steady state value of x is beta over alpha, say. So, and then as beta gets bigger, the output, as beta, the input gets bigger, the output gets proportionally larger. What happens in cooperative systems is that in the ideal case, there's a threshold, that if the input is below threshold, then there's no output. And then if the input is above threshold, then there is an output, and often in an idealized system, the output saturates. So you go from having zero output to full output with a very sharp transition. So that is an idealized cooperative systems. In biological systems, instead of it being a sharp transition, it's a smoother transition. And then on the high end, maybe the output maybe still continues to rise a little. But for the analyses that we'll do, often the threshold type behavior is a good approximation. So what that means in our case is that in biological systems, there can be noise, or essentially, a little variation. So what can happen is that even if the pathway isn't activated, a little bit of the transcription factor might get phosphorylated and go into the nucleus because of, people say, pathway crosstalk or some other events. So there might be some level of transcription factor in the nucleus that is smaller than the level that you would have with full activation, but still every now and then the transcription factor gets in there. So that is sort of in this regime of the pathway. And in a linear system, that would be enough to activate everything downstream. So say that this is, a proliferation type pathway, that a transcription factor going into the nucleus says that a cell should start dividing. That is not always good. So cells dividing uncontrollably is one of the phenotypes of cancer. So the cell wants to protect against low levels causing activation of a pathway that could be deleterious under a certain environment. So evolution seems to have picked cooperativity as a standard theme for having a pathway that doesn't respond to low levels of input. Inputs have to be above a threshold level in order to get an output. So we will talk about those mechanisms. All right. So now we are going to look at how much information it takes to turn on a specific gene. So here's our model now. We have a pathway that's been activated, and the cell wants to use this pathway to turn on a specific gene. So often a signaling pathway doesn't turn on a specific gene, it turns on a whole number of genes that then all work together in a second pathway. But to simplify the picture, we'll think about a transcription factor that's going to activate a single specific gene. So here's our gene encoded on DNA, and here's the transcription factor. And the way that yes?
When you said information earlier, were you talking about information in the sense of information theory?
Yes.
Okay, so are we covering that?
We will today. Yes, we'll even take a log. All right. So yeah, so we'll use what we need. All right. So the way that transcription factors recognize genes is they are sequence specific. So a sequence specific transcription factor just means it's one that recognizes a specific DNA sequence. So genes upstream of the protein coding region have a promoter, and the promoter includes specific sequences called regulatory elements that are recognized by transcription factors. They're called regulatory elements because they regulate the expression of the gene. There are also regulatory elements far away from the protein coding region called enhancers. So way far away, there are enhancers that transcription factors also bind to. And even though they're far away, if you think about the primary sequence of DNA, what happens is that they wrap around in three-dimensional space. So a transcription factor will bind here, but then sort of cause the DNA to loop over and come into contact with the promoter and then activate the gene. So all of those details are the subject of research these days, identifying enhancers and relating them to genetic variants that affect complex traits in disease. So that's sort of active research figuring out how all of that works in specific cases. But in general, that's how transcription factors work. So some are sequence specific. There are also non-sequence specific transcription factors. So the idea is that in order to transcribe a gene, you need a lot of apparatus. You need something that tells you which gene to activate, but then you need all the machinery of RNA polymerase, and RNA polymerase is a big complex that has to get recruited. So those are non-sequence specific transcription factors that a transcription factor will recruit. So often a sequence specific transcription factor will have one domain that's called a binding domain that recognizes a DNA sequence, and then another domain called an activation domain that sort of pulls down the rest of the machinery that's needed to transcribe a gene. Okay, so here's this transcription factor. We want it to turn on a single gene, and the question is, how many base pairs does it need to bind to in order to turn on the gene? So how big is this binding width? So we'll call it W. So how big does the binding, the recognition sequence have to be in order to turn on the gene? So the way that we'll reason through that question is what we're going to say is we don't want that motif to show up anywhere else in the genome. So we have this motif that a transcription factor binds to. So let's just say the transcription factor binds to that sequence of W nucleotides or base pairs. Wherever it sees it, we want that sequence to occur just upstream of our gene and nowhere else. So the question is, how often does this sequence occur by chance in the rest of the genome? And we want that number to be small. So, so how often does a motif, and a motif just means a nucleotide sequence of length W appear in a genome? And we need to know how big the genome is, so we'll call that G. And our idea is that binding widths have evolved to be big enough that that sequence is unlikely to occur by chance anywhere else. So we want that sequence to be unique if the rest of the DNA is random. So this part of the DNA, let's say, has sort of co-evolved with the transcription factor, that it's recognized by the transcription factor. The rest of the DNA in the genome, we'll just say, is random. How big does the motif have to be that it's not going to occur anywhere else? So our model is going to be that our genome is going to have independent nucleotides that occur independently at each position with uniform probability. So that at each position we're equally likely to have an A, T, C, or G. Yes?
Is that actually true in reality?
So It is not true in reality, but it is close enough to truth that we won't really make any big errors in doing it. So, excuse me?
Pyrimidines and pherines are both equally probable when you're analyzing DNA.
So, for a purine on one strand, you need to have a pyrimidine on the other strand. So that was one of the first discoveries about DNA that got people interested in a molecule, that the amount of A always matched the amount of T and the amount of C always matched the amount of G. Like, that was a clue. So what people look at, though, the one thing to look at is AT versus CG content. So some genomes are a little more AT rich, some are more CG rich. If you remember from molecules and cells, one of the big differences between AT and CG is that AT has 2 hydrogen bonds and CG has 3 hydrogen bonds. So CG is a little tighter. So it turns out that I think organisms that like to live places where it's really, really hot tend to have more CG nucleotides in their genome to make it a little more stable. Malaria is a disease. The parasite is called Plasmodium, like falciparum or something like that. That genome is really odd in having a lot of AT, which made it very, very hard to sequence because there are long stretches of the same nucleotide. So in some genomes, there's a bias to a little more AT or a little more CG. In some regions of our own genomes, there's a little more AT bias or CG bias. In protein coding regions, the nucleotide frequency is really pretty close to uniform. There's some regions in the genome, though, where there are like stretches of A's or stretches of T's. Often that's from retrotransposition, where a transcript with a poly A tail gets reincorporated into DNA, and then you see a low diversity region. But for the most part, it's a good approximation. So each position identically, independently distributed. So uniform A, T, C, or G. So now we're going to look at a specific. So now we have our big genome G, and we're going to lay down our motif of length W, and we're going to say, what is our probability of a match? Yep. So our probability of a match is for each position W, there's a 1/4, and since they're all independent, it's 1/4 to the W. 1/4 to the W. So then what we do is, all right, so is everybody okay with that? All right. So then what we do is we move the motif, we slide it over one position, and then we ask, what's the probability of a match at this next position? And you might think that you have to worry about whether or not there was a match at the previous position, because that sort of, locks in what the nucleotides are. But in reality, we're just looking at the single point function, so we don't even have to worry about that. The probability of a match at the next position is also 1/4 to the W. So at each possible starting location, the probability of a match is 1/4 to the W. So then what we want to do is we want to calculate, well then, how many matches do we expect? So we'll say that K is the number of matches in the genome. So what we're doing is G total experiments. So out of G possible starting positions, we have K matches. And then if we call small p the probability of a match, we have small p to the K, 1 minus small p to the G minus K, where this is what we're calling small p. So a binomial distribution. Yes?
Go over that again. What was G? What was K?
All right. So G is the total length of the genome. Like, a good hint is that genome starts with G. So G is the total length of the genome. K is the number of matches. So we want K to be small compared to the genome size. Like ideally we want it to be 0, but because these are random variables, they can't, like, it's never going to be 0. There'll be some probability that it's bigger than 0, but we want that probability to be small. So
Wouldn't you want at least 1 for the 1 gene?
so we're leaving aside the one
So it's not-
that we've reserved,
Not considering
not considering the one that has co-evolved with it. Yes.
if it's 0, then it's not. If it's really 0, it's not.
Yeah, we want like that. So those W bases we're leaving off to the side because evolution, let's say, has fixed those positions. The cell will die if that gets changed. It's co-evolved with the transcription factor. Everywhere else, so really the other G minus W possible starting positions. But because the genome size is much bigger than the motif size, we don't have to worry about that. We'll still use capital G as the length of the genome. This will, sort of, it's an order of magnitude type calculation. So we want to get the width, like we're going to use this to figure out what the length has to be, and we'll really get it to like plus or minus 1. Like this is enough to plus or minus 1. Because if you think about these probabilities, plus or minus 1 in the width gives a factor of like 4 to 16 in these binding in the match probability. All right. So now any issues with this binomial probability? Yeah, all right. So here's what we're going to do. So 1 minus p for small p is about equal to e to the minus p. If you do a Taylor series of e to the minus p, the first two terms are 1 minus p. So then plus order p squared. And p is so small that p squared we will ignore relative to p. So we replace this with e to the minus p times g minus k. And so we could write this as e to the minus p times g and then 1 minus k over g. So just factoring out the g. And then we want to be in the situation where we don't have many hits. If we have a lot of other hits, we're in trouble. So we want k to be much less than g. So if we got hits on the order of the genome size, then this transcription factor would be binding everywhere. So we don't want that. And so we'll ignore terms of order k over g also. So we'll ignore that, or you think about, we're working in the that k over g goes to 0. So this is equal to e to the minus p times g. And we're going to call p times g lambda because everybody else does. So we'll call that lambda. All right. So what p times g conceptually represents is p is the probability of getting a hit per position, and g is the total number of positions. So p times g ends up being the average number of hits that we expect. All right. So like already you get the idea that we want p times g to be small, but we're gonna call it lambda. We could have called it like n bar or something like that, but then that gets, everyone else calls it lambda, so we'll do that too. Oh, that's my phone. All right. So I have a question. I updated my iPhone operating system, and like the next day, my phone wouldn't start, and I had to restart it from iTunes. So like, anyone else have that problem? No?
I just don't upgrade.
Wait, you just don't update it?
No.
I know, I shouldn't, but I did. Yeah, all right. So it's like first world problems, like my iPhone won't turn on. Okay, so now we have to, so now we're going to do this combinatorial factor. So if you remember what these are, on the bottom it's 1 times 2 times 3 all the way up to times k. So that's k factorial. And on the top, every time you have one of these, you have like g, and then g minus 1, g minus 2, all the way up to g minus k plus 1. So here we have k terms. So that's the standard combinatorial factor. And then what we're going to do is the same way that we factored this factor of g out of here, we'll factor the factor of g out of all of these. So this is going to be g to the k times 1 minus 1 over g times 1 minus 2 over g times all the way to 1 minus k minus 1 over g, and then k factorial. And then the same way that we ignore terms like k over g, we're going to ignore all of these. So what we get is g to the k over k factorial, and then we put that back in here, and we get that the probability of k hits is equal to g to the k over k factorial p to the k, so that's that p to the k, and then e to the minus lambda. So that's lambda to the k over k factorial e to the minus lambda, the Poisson distribution. So we get a Poisson distributed number of hits, which is really, what we should have expected, because the Poisson distribution is a limit where you have an experiment that you repeat lots of times with a small success probability per time, and the limit that the success probability goes to 0, the number of trials goes to infinity, and the product is a finite number. You get a Poisson distribution with that as the mean. And so that's what we have, a mean with lambda hits, where lambda is equal to 1/4 to the w times g. All right, so there are some questions. Yeah?
Do you remember what you did in the upper right corner there?
What I did in the upper right corner is I ran out of room. So here's what we're doing. So you're comfortable with the numerator for combinatorial factor? All right. So what we're going to do is we notice there's a big capital G in each of these factors, okay? So g times g minus 1 times g minus 2 times all these others, all right? So a very common thing to do when you're doing a combinatorial factor and this number on top is much bigger than the number on the bottom, like much, much bigger. So here it's much bigger because our genome size is going to be like millions or billions. And this is going to be like a small number of hits, like 1 or 2 or even 100, okay? So when the top number is much, much bigger than the bottom number, we're going to factor out that big top number out of each of these terms, okay? And there are k terms because, it's choose k, so we have a k factorial on the bottom, and we know we've got one on the top for everyone on the bottom. So we factor out that factor of g from each of them, so that's k powers of g. And then if we factor out a g from the first term, we'll have 1 minus 1 over g. And if we factor out a g from the second term, we'll have 1 minus 2 over g. And it goes on like that. So that's all of these terms. 1 minus 1 over g, 1 minus 2 over g. All right? You okay so far? All right. And then all of these terms, like 1, and then 1 minus 1 over g, 1 minus 2 over g. We're going to say that all of those are so close to 1, we're just going to put 1. Because g is so much bigger than the numerator that a small number over the genome size might as well be 0. So that's what we're doing. All right.
Still overestimated by a small amount when you put that substitution in there.
So you mean in doing this?
Yeah.
So we have much bigger problems than this approximation. We want to get to the order of magnitude. So nobody's asked me about repetitive sequences. So we have something called ALU sequences. Eukaryotes have been repeatedly invaded by viruses that cause retrotransposition, which means that a virus gets into your DNA and then encodes for proteins that lets itself get transcribed and then reinserted other places in your DNA. So I think like 25 to 50% of our DNA is like really this stuff, that it's the same sequence everywhere. So really this genome size, if we think about the size, like our high information content DNA that follows this random pattern is like only half of the size of our DNA. Plus, like we talked about histones, our DNA is packed into two general forms, euchromatin, which is transcriptionally active, and heterochromatin, which is silenced. And one of the mechanisms for silencing is that just physically it's unavailable for transcription. It's packed up into like some big messy ball that can't get unwound. Like, if you give a ball of yarn to I don't know, I didn't have cats, but like I've seen things. So like, a cat playing with a ball. So a lot of the genome isn't available. Also, the genome has big regions that are also like the same sequence just repeated. So for example, the centromere of DNA is a very low complexity sequence. It's just, I don't remember exactly what the sequence is, but it's very low complexity, so it doesn't behave with this random pattern. And telomeres, so the ends of DNA, it's a 4-nucleotide sequence that just gets repeated forever. And, so then enzymes like telomerase, that Carol Greider won the Nobel Prize for, maintain the lengths of telomeres, but it's just a random, it's not random, it's the same sequence over lots and lots of nucleotides. So there are a lot of regions of the genome that are low complexity. And if we think about like the real number of trials we have, it's not the genome size, maybe it's the genome size divided by 2 or 10 or something else. So in the end, even an order of magnitude of 10 in genome size that we're off won't affect us that much. So also, so while we were discussing this, so who knows the birthday paradox? So what's the birthday paradox?
That you can have a small number of people in the room, but the probability of 2 people having the same birthday is actually quite high.
Yeah, so what's I forget what the number of people you need in the room to have like a 50% chance to have like one pair expected on average? Like
It's around 10,
30? It's 23.
So we won't make this a homework problem. Yeah, my memory was actually close to 30, but I would buy 23. I guess it depends whether it's a leap year or not.
Because everybody chooses.
So the way for solving that problem actually is you go through this step, but then instead of ignoring these factors of like 1 over g, where g would be 1 over y, where y is the number of days in the year, you take the lowest order contribution in 1 over y, which becomes something like k times k minus 1 over 2. Like you're summing up 1 over y plus 2 over y plus 3 over y plus, k over y. And then you sum this up, there's like k times k minus 1 over 2 over y. And then that has to be close, that has to be the number of people. So it would be something like, solution would be the number of people squared is 700. So the number of people you need is square root of 700. So that's 10 times the square root of 7. And the square root of 7 is, well, since someone said 23, I'm guessing the square root of 7 is close to 2.3.
I'd say 2.7.
All right, great. That's not in the notes, nor will it be on a homework. All right, so back to the problem we started with. But since we're discussing that, I will say that 2 of my kids actually have the same birthday, 3 years apart. And that birthday was July 4th. And when the first one was born July 4th, I learned something very important about hospitals that many of you will learn, is that the new residents start on July 1st. So we had an entirely unexperienced team helping deliver the baby.
Twice.
So the second time we were ready for it. Actually, the second time, like it was that morning, I was busy putting together the presents for the kid who was about to turn 3, and I was like, and it was like, anyway, like I think you have to start putting away, stop putting together the presents. It's time to go to the hospital. So, and she was actually, I think, like in transition in the car on the way to the hospital. I don't recommend that. All right. Okay. So we have probability of k hits is equal to lambda to the k over k factorial e to the minus lambda, where lambda is equal to the average or the mean or the expected number, which is 1 over 4 to the w times g, where g is the length of the genome. So then, just as a rule of thumb, let's say that we want this to be less than 1. We want, less than 1 hit on average to give us a ballpark figure. So then we know that sort of, widths that are bigger than that will work really well, because then we'll have like maybe only a 25% chance of having a hit. And widths that are shorter than that will not work well at all because we'll have too many hits. So this is sort of the critical value that we want 1/4 to the WG is we want it to be less than 1, or we want 4 to the W to be greater than G. And then we'll take the log of both sides. And so the thing is, we're only allowed to use 3 bases for logarithm. We're allowed to use base 10. We use that for p-values and for genome sizes. We're allowed to use natural log because it's natural log. And we're allowed to use log base 2 because we think in binary. So since there's a 4 here, we'll take log base 2. So, and then the log base 2 of 4 is 2. So we get 2w is greater than the log base 2 of g, or we need w to be bigger than 1/2 log base 2 of g. Okay. So in terms of information theory, like we think in terms of bits of information, the number of bits of information that the motif has to provide is the log base 2 of the genome size times 1/2. So now we'll think about some typical genomes and what this says about the size, the length of DNA that has to be recognized. So we have virus, single cell, so prokaryote, like E. coli, bacteria, yeast, and then metazoan. So metazoan is just a multi-celled animal. And so, things like worms, flies, mammals. Okay. So I think so what do I use in the notes? So viruses, so the genome size, 10 to the 4th for a virus. Yeast and bacteria are 5 to 10 megabase genomes. So mega is 10 to the 6th. So this will be about 10 to the 7th. And people are 3 times 10 to the 9th. So we'll round up to 10 times 10 to the 9th or 10 to the 10th. And then the binding width we need is we'll just take 1/2 log 2 of 10 to the 4th. So that's equal to 1/2 times 4 times the log 2 of 10. And the log 2 of 10 is about 3.3, so I'll write that as, say, 10/3. So we get 20/3, which is about 6 to 7. So a virus that wants to have a transcription factor that recognizes just the virus has to be 6 to 7 base pairs. However, like really the virus is replicating itself in a cell, so it's probably more the host genome size that's important for the transcription factor size of a virus. But just in case, there it is. So so here for single cell species, so we get 1/2 times 7 times 10/3. So 70 divided by 6. So that's about equal to 12. So 12 base pairs of recognition is about enough for a bacterial cell. And then for humans, 1/2 times 10 times 10/3. So that's equal to 50 divided by 3. So that's about equal to 17, something like that. So we need about 17 base pairs of recognition is the trade-off between recognizing way too many locations at random and being able to specifically pick up a single gene. So actually, many of you have done PCR amplification or real-time PCR, quantitative PCR with primers. So when you design primers for PCR, usually your primer length is like 20 to 25 base pairs. And that comes from the same reasoning that that's the length you need to ensure that you're only amplifying what you want, that your primers aren't going to hit somewhere else in the genome by chance. So, this is the reasoning for primer length. It's also a guide for transcription factor recognition. So now we're going to take a look at how transcription factors recognize those base pairs. So we'll start at the projector again. All right. So any questions on all that stuff? Yes.
So, great. My question is, how does this relate back to what we were talking about at the beginning with cooperativity and saturation? So does this have to do with like the threshold or how things are set?
So we'll get to cooperativity. So what we'll see is that to recognize this many base pairs of DNA, we need transcription factors to cooperate. Okay. So that's the answer. So we're going to see how transcription factors recognize DNA. So look at some pictures. Oh. What happened to those pictures? Where are they? Oh, here we go. So here is a picture of a typical transcription factor. Let's see, 3D view. All right, great. So here we go, 3D view. All right. So there we go. That's a great view. So here's what we're looking at. So I forget what the name of this transcription factor is. It's probably better seen in the dark. There we go. All right. So you can see in blue is the DNA that the transcription factor is bound to. And then so now there are two features to look at. First are these two pink alpha helix tubes. So if you see what they're doing, they are sitting inside the major groove of the DNA. You can also see they're sort of distorting the DNA a little bit. Oh, yes?
Can you
So I don't have a switch here for those. Is there, like, someone want to try that switch at the back of the room? Volunteer? I need a volunteer. Is there a switch at the back of the room? There's like a plate there. Is that a switch? Yay! All right. So the answer is yes, we can turn off those lights. All right. So there's the DNA in purple, and you see, the bases and then the backbone. And so these two alpha helices that are sitting inside the major groove of DNA, so like DNA has the major groove and then the minor groove over there, there are side chains that are not being drawn because it would just be too cluttered to look at, that are sticking into the major groove and making hydrogen bonding contacts with the nucleotides. So usually we think about DNA as a double helix where hydrogen bonds hold nucleotides in one to nucleotides on the other. Actually, recognition of DNA sequences by transcription factors works the same way with hydrogen bonding. And for some families of transcription factors, people have actually worked on recognition codes that say that if you want to recognize a specific DNA base pair or a nucleotide, this is the residue that you would need at the alpha helix in a specific position. All right, so these two helices are responsible for recognition. And if you remember DNA structure, that sort of standard DNA, one full turn of the helix has, well, you could count them, it should have about 10 residues. So, or 10 base pairs, 1, 2, 3, 4, 5, 6, 7, 8, 9, something like 10. So there should be 10, but the alpha helix can't really make contact with all of them. It can only make contact with the ones that are sort of at the bottom of the well, so about 6 at most. So each alpha helix can recognize 6 base pairs at most. Sometimes they're a little wobbly, and it's really more like they're recognizing 4 or 5, which means that in human, we need at least 3 to 4 alpha helices to recognize enough base pairs that we're able to turn on a specific gene and really not turn on any other gene at random. So we'll need multiple alpha helices to come together. So you see in this crystal structure, there are 2 alpha helices recognizing a motif of probably about 10 to 12 base pairs, where it's recognizing a little bit over here and a little bit over here, and then in the center, it's not really recognizing anything. So motifs can be like that there can be a gap in the middle where really anything can be there, but it's recognizing this piece and this piece. So the other thing to notice is that there are these 2 other alpha helices that are sitting right above the DNA backbone. So what's going on there is that in addition to recognizing DNA, transcription factors have to sort of like sticking to DNA. And one mechanism for recognizing is they sort of stick to DNA and then they slide up and down the double helix until they find some place that they really, really like. So these alpha helices are usually have positive residues on them so that they interact favorably with the negatively charged phosphodiester backbone. So usually a lot of lysines and arginines. Actually, histones also have a lot of lysines and arginines. And I think this one is a zinc finger transcription factor. And what that means is that there's a zinc atom here that helps keep this whole transcription factor structure together. So, so that is an example of a transcription factor. So we'll go back, we'll take a look at a different one. So phos-Jun leucine zipper. So phos and Jun are a heterodimer. So here we go. So let's see, Control Shift plus. All right, so we'll try to. There. All right, so here's a picture. So the way leucine zippers work is, so the leucine zipper part are these two big alpha helices. The leucine part are that there are leucine residues on the two alpha helices where they come together over here. Leucines are hydrophobic, so it's more favorable energetically for these two alpha helices to bind to each other than to be solvent exposed. And then that gets like the business end, the sequence-specific recognition end, positioned to hold the DNA like a pair of chopsticks or pincers or whatever other simile you want to use. So this is where the DNA is recognized, and then this part is the structural stability. This is a heterodimer. So these are two different transcription factors that are cooperating to recognize DNA. So this is where cooperativity comes in, that if one binds on its own, it has the favorable interactions with the major groove, but it has unfavorable interactions because it's solvent exposed. Then when the other one comes in, it gets favorable interactions with the DNA, plus it's more favorable than the first binding because now they can get their leucines next to each other and have more favorable energetics. So the second one binding is more favored energetically than the first one binding, and there can be cascades like this where each time you add one, it's sort of more and more favorable. So like with the standard example of hemoglobin and cooperativity, where every oxygen bound makes the next oxygen more favorable to bind. Here it's sort of the same thing, that once you have one of the transcription factors bound, it makes it more favorable for the next one to bind. Let's see, so what time is it? So we have time for one more picture. So the last picture is beta sheet. So this is P50, P65. So these are important, I think, cancer-related transcription factors. And where are my navigation tabs? 3D view, great. So here, so there's the DNA in the center, and then this big sort of messy thing. So here, the recognition, so you can sort of see this part of the protein making responsible for recognition. So here, it's not an alpha helix, it's a beta sheet. So another type of structural unit. But it still is the same general idea that it is a protein structural element that is making contact with the major groove, and by making contact with the major groove, it gets about 4 to 6 base pairs of recognition. So those are the pictures. So we can turn the lights back on. We can turn the one in the back on too, I guess. So what we've learned is that from information theory, we've learned that for people, we need about 16 to 18 base pairs of recognition. That each recognition unit, so recognition units, structurally, based on the structure of DNA, recognize 4 to 6 base pairs. So we need multiple recognition units to come together and cooperate in order to recognize DNA. So the next lecture, we will work through cooperative models for transcriptional activation. Have a nice weekend.